Barbara Melchert, and I'm from the Department of Molecular Pharmacology of the University of Groningen. Today's date is March 27th, and this is a recorded class for the course Pathology and Immunology for first-year pharmacy students. Now, in the former classes, we talked about the innate immune system and adaptive immune system, which cells belong to what, how do they grow up, where do they grow up. And this class will be about how B cells respond to a threat, how cytotoxic T lymphocytes, CD8 positive T cells respond, and how they are being helped by T helper cells. So here is an overview of what we have been talking about before. The innate immune system is the fast part of the immune system. It reacts fast. It consists of epithelial barriers, of phagocytes that are present in these barriers. In the blood, we have the complement system that protects us. And if they cannot handle it, then they will get help from the adaptive immune system, which consists of lymphocytes, B lymphocytes and T lymphocytes. B lymphocytes can make antibodies that can help, and T lymphocytes can become effector T cells that can help out in various ways. And that will be discussed in this class and some more that follow this class. So responses of lymphocytes can be divided into either B cell responses or T cell responses. If a B cell recognizes an antigen with its B cell receptor, it becomes activated, it becomes a plasma cell, or generally known as an effector cell, and it will secrete antibodies. Also, some memory cells will develop that will initiate more rapid secondary responses when you're exposed to an antigen again. And the plasma cells that secrete antibodies can also become memory cells that are long-lived and can continue a supply of antibodies for the rest of your life. Within the lymphocytes, we also have the natural killer cells that have a lymphocytic origin, and those are necessary to destroy virus-infected cells and cells that have been antibody tagged, but they're actually considered to be part of the innate immune system and have been discussed in that context. Then T cells. T cells either become cytotoxic T lymphocytes characterized by expression of CD8 that can kill cells that are either infected with a virus or tumorous. They can become T helper cells that are characterized by CD4 and that can help other cells do things. Or they become regulatory T cells that can keep immune responses down, suppress immune responses when they're not really necessary. So what does an adaptive immune response look like in general? Well, it starts with the recognition of the antigen here by a naive B or T cell. That cell becomes activated and starts dividing. We call this phase clonal expansion, so the number of cells increases. Then they develop into effector cells and start up an effector response, so they actually start to be doing something, and that takes around 7 days. Then when the antigen has been discarded, has been removed, then all these cells are not necessary anymore. The effector cells die off, and only a few memory cells remain, and the system goes back to homeostasis. Another way of depicting it is here for T lymphocytes. Here's an antigen presenting cell that is presenting antigen in the context of major histocompatibility complex molecules, either recognized by CD4 positive T cells when it's MHC class II, or by CD8 positive T cells if it's MHC class I. They become activated when they recognize something. They start dividing, you get clonal expansion, and then they differentiate into effector T cells, and some will become memory T cells. So the effector CD8 positive T cells, or the cytotoxic T lymphocytes, they go out and look for the infected cells or the tumorous cells and kill them. And the CD4 positive T cells, they start helping other cells with whatever function they have. So macrophages with killing phagocytosed bacteria, for instance, B cells with making antibodies, and CD8 positive T cells with killing things. Here in more detail how CD4 positive T cells can help other cells. So here we have the B cell that becomes activated, becomes a plasma cell, starts producing antibodies. This cell can be helped by CD4 positive T cells. I'll come back to that later again. Here's the macrophage that has phagocytosed bacteria, for instance, and is being helped by the CD4 positive T cells, killing those. And here's a CD8 positive T cell that recognizes an infected host cell that needs to be killed, and that one can also be helped by this T helper cell. Well, a little bit more about the B cells. When a B cell recognizes an antigen with its B cell receptor, and it's the first time it does this, you get clonal expansion and the generation of plasma cells that start producing antibodies. If you and there are some memory B cells that will develop. If you look at the time frame, then when you're first exposed to the antigen, it takes about 2 weeks to get a peak in antibody concentration. We call this the primary immune response. And these antibodies consist of IgM antibodies. Remember the big molecule of 5 antibodies together, which is called the IgM. But these B cells can get help from T cells. Here, the B cell that produces IgM when it's activated, but it can actually get some help from CD4 positive T cells. So the CD4 positive T cells is activated by an antigen presenting cell, and it helps the B cell. And what does it help the B cell with? With becoming memory cells, and with switching its antibody production. So normally a B cell makes IgM, but when it gets help from the CD4 positive T cell, it can start producing IgG, IgA, or IgE, which it normally cannot. And that also means that when you're exposed to the antigen for a second time, the more effective memory cells can respond a lot faster to that antigen than during the primary immune response. As you can see here, if a memory cell that developed after contact with a CD4 positive T helper cell sees the antigen again, it becomes a plasma cell and starts producing, for instance, IgG antibodies in a much faster way than it could before. So now you already have substantial amounts of antibody in the first week, and it can also produce a lot more. So what were the types of antibodies, or immunoglobulins as they're also called? Again, we had IgG, IgE, IgM, and IgA, with IgM being a pentamer of 5 antibody molecules, IgA being a dimer, and IgG and IgE being monomers. And the difference between IgG and IgE is in this tail that is slightly different and therefore can bind to different receptors on cells. So what can these antibodies do? Here's the B cell, it recognizes an antigen, it becomes a plasma cell, starts producing antibody. And what these antibodies can do is neutralize toxins by binding to them and preventing them from entering cells or binding to receptors. It can induce complement lysis, so if this is a bacterium that has antibodies attached to it, it can activate the complement system, and then the complement can make these membrane attack complexes that make pores in this bacterium that will lead to lysis of the bacterium. The coating of a bacterium with antibody can also lead to opsonization, and that is basically phagocytosing by macrophages. Macrophages recognize this tail of the antibody, and that leads to phagocytosis of the bacterium. Or when cells are being coated with an antibody, the natural killer cells, NK cells, can be activated, and they will start killing this coated cell, and we call that antibody-directed cellular cytotoxicity, or ADCC. Which antibody can do what? IgG is the most versatile antibody because it basically can do everything. IgE can only activate mast cells and eosinophils. IgM can activate the complement and neutralize things. And IgA is also very important for neutralization, and is only present at the mucosa, so it's very important for mucosal defenses. So how do CD4 positive T cells help cytotoxic CD8 positive T cells? Well, here's a CD8 positive T cell that becomes activated because it recognizes an antigen presented by the antigen presenting cell in MHC class 1. It starts to divide and proliferate, and then it goes out into the tissues to look for the tumorous or virus infected cell. If there are CD4 positive T cells around, they can help the CD8 positive T cell to become a more competent cytotoxic T lymphocyte. So they help with memory building, and they help with improved division of the cells, improved cytokine secretion. So they make the immune response more effective. And a disease in which you can see how important this CD4 positive T cell help is AIDS, which is caused by the virus HIV, human immunodeficiency virus. This is similar to the coronavirus that we're currently under lockdown for. The coronavirus, the official name is SARS-CoV-2, is the virus, and the clinical disease it causes is COVID-19. So it's similar to HIV. HIV is the virus, and it causes a disease which is called AIDS, acquired immunodeficiency syndrome. And it's caused by HIV because HIV targets CD4 positive T cells. Well, the initial infection can be influenza-like, so you have flu-like symptoms. And then the virus can hide for on average 8 years. And then something happens which gives you overt disease, and that's when the CD4 positive T cell count comes below a certain threshold of 200 cells per microliter. and then you lose your effective adaptive immune system, and then you're going to suffer from infections and tumors that will eventually kill you. So how does HIV do this? So this is the HIV particle, which specifically has proteins on the outside that specifically recognize CD4, and can then bind to a chemokine receptor called CCR5. So here we have the viral particle binding to CD4 and then binding to the chemokine receptor, and then the RNA cargo of the virus can be injected into the cell, into the CD4 positive T cell. So HIV is an RNA virus again. This RNA will generate viral DNA through reverse transcriptase, synthesis. This DNA then incorporates into the host DNA, and this host DNA is being read and being replicated, so you get a lot more RNA molecules, and the proteins of the virus will be produced as well, and that will lead to reassembly of the virus, and then you'll get release of lots of virus particles. If that happens, the cell is then killed. So you're going to lose your CD4 positive T cells then. Here is shown what is happening. Here, this is a mucosal surface where the virus initially will enter, and will then recognize CD4 positive T cells and will infect them at the local mucosal surfaces. But there are also dendritic cells there that will eat the virus and transport it to the lymph nodes. Of course, the lymph nodes are the reservoirs of CD4 positive T cells, so it's like going to a party for this virus. There's lots of virus around, lots of cells around that it can infect. So these lymph nodes become a reservoir of the virus, and also it will help the virus spread throughout the body. And of course, you have CD8 positive T cells that will try to fight the virus, and they manage to do a good job. So then you get partial control of viral replication, and you get clinical latency with the virus being hidden in lymph nodes with only low-level virus production. But then when your immune system is stressed, for instance because of other microbial infections, then the virus will reactivate, you get increased viral replication, and then your lymphoid tissues will be destructed, and you'll completely lose CD4 positive T cells, which means your CD8 positive T cells won't be very effective anymore, and then you'll die of opportunistic infections or tumor development. Here, what it looks like in patients. On this axis you see the CD4 positive T cell count in blue, and here is the number of viral particles of viremia in yellow. So during the primary infection, the acute phase, you'll see the number of viral particles going up and the number of CD4 positive T cells going down. Then you get the situation of clinical latency, where you sort of have a somewhat lower but constant level of CD4 positive T cells, and the number of viral particles stays the same. But then something happens for the virus to really activate, then the number of viral particles goes up again, and you completely lose your CD4 positive T cells, which will eventually lead to death by opportunistic infections or tumors. So we talked about B cells now and CD8 positive T cells. How CD8 positive T cells help out in a viral infection is explained in my class about SARS-CoV-2. So for more info, have a look there. And in the next classes I will talk about how CD4 positive T cells help other cells.